Hello, my name is Jackson Scott, and I am a third-year biochemistry student at the University of Calgary. This summer, I had the privilege of working under Dr. Wee Young and Dr. Atif Raipur at the Hotchkiss Brain Institute on my summer research project, which was identifying novel targets in oxidative stress pathways in demyelinated lesions. Now, by demyelinated lesions, I refer to the disease condition caused by multiple sclerosis, which is a chronic neuroinflammatory disease caused by an autoimmune response primed against axonal myelin sheaths. As you can see in the bottom right, we have a healthy neuron with a thick myelin sheath. However, in MS, you get the degradation of this myelin sheath and ultimately neuronal death. You can see this on the left with these demyelinated lesions in the human brain and central nervous system. Now, the reason this is so important is because Alberta has some of the highest incidence of MS in the world. Causes for MS remain variable and ambiguous, the disease progression is complex with many different forms, and approved treatment options are limited, especially in primary and secondary progressive MS. Oxidative stress is the accumulation of reactive oxygen and nitrogen species within tissue. Oxidative stress has been heavily implicated in MS disease severity and progression and has become recently a promising area for future therapeutics. Prior to my project, my supervisor, Dr. Raipur, did RNA sequencing analysis of 2 animal models of multiple sclerosis, experimental autoimmune encephalomyelitis, and lysolecithin injection, as well as examined human MS public datasets for differentially upregulated pathways and genes associated with disease. Upon settling upon myeloid cells as her focus with macrophage and microglial immune cells, bioinformatics pathway analysis was performed and found that oxidative stress pathways were upregulated in the disease conditions and that certain genes were associated with it. For this project, we focused on the 3 apolipoproteins, C1, C2, and E as the genes we would investigate. The model we used was the lysolecithin injection model. The lysolecithin injection model is an injection of detergent into the spinal cord in this case to simulate the demyelination, immune response, and then remyelination aspects of multiple sclerosis. It was chosen because it has a predictable demyelination peak occurring at 7 days post-injection. The gaps in knowledge we hope to address are that ApoC1 and ApoC2 have been unexplored in the central nervous system, the 3 apolipoproteins have yet to be described in MS, and how the 3 proteins connect to oxidative stress pathways is not known. Now, we hypothesize that the 3 apolipoproteins are highly expressed in lesions of LPC-induced demyelination, and that their expression will change in animals since ApoE is an age-related gene and the other 2 might be as well. Our first aim was to evaluate the temporal expression of the apolipoproteins in LPC model. We did this by taking LPC tissue and staining it immunohistochemically for our target proteins and astrocyte cell markers and immune cell microglial macrophage cell markers, that being GFAP and IBA1 respectively. These stains were then analyzed using an SBA confocal microscope and put in a 3D imaging software called Amaris, which allowed the signal to be turned into 3D structures and analyzed, as you will see shortly. The data from Amaris came back here, where we can see the total number of double positive cells per lesion area and also the percentage of double positive cells. We can see that ApoC1 has a very high correlation with astrocytes, a medium correlation with immune cells, but as demyelination worsened from 1 day to 7 day post-injection, there is an increase in ApoC1 expression. ApoC2 yielded no significant results. This is Amaris, where it takes the immunohistochemical stain, the fluorescent stain, and allows surfaces to be made to see the overlap between GFAP cell body marker and the target protein. This hints strongly that this astrocyte is located and potentially expressing the target protein. This is where the data comes from. Once again, ApoE did have significant results and saw an increase in expression as demyelination worsened. However, note it is very lowly associated with immune cells and still moderately associated with astrocytes. For aim 2, we aim to look at the apolipoproteins across different ages in mice. ApoC1 and ApoC2 had no differences across different age groups in mice. However, ApoE, the only known age-related gene, did, as younger mice had a higher expression of ApoE compared to older mice. Once again, though, the astrocytes show more expression than the immune cells. So in terms of significant results, ApoC1 and ApoE seem to have more expression at peak demyelination by both astrocytes and immune cells. Astrocytes are highly correlated with all 3 apolipoproteins. ApoE seems to be predominantly extracellular, while ApoC1 and 2 are largely associated with individual cells, and the age-related findings to ApoE warrant further investigation to determine if there's truly an age difference. I'd like to thank Dr. Wee Young and Dr. Atif Raipur for giving me the opportunity to do this research, and Alberta Innovates for funding this project. This is my literature cited, and thank you so much for listening to my presentation.